Hello, BISC1323. This is the beginning of recorded lecture 5.2, and we are still in the circulatory system chapter, although we're going to finish it up in this one. So we began this chapter and talked about the process of circulation, specifically within humans. And we saw, steps 1 through 8, all that stuff in the last one. I want to point out now that we can divide this whole process into two main circuits. It's kind of like a figure 8. The first circuit is called the pulmonary circuit, which goes from the heart to the lungs and back to the heart. And the other circuit is the systemic circuit, which goes from the heart to the head and the body to deliver oxygen and then back to the heart again. So this is referred to as double circulation because of these two main circuits. The key term says the flow of blood in two main circuits, the pulmonary circuit through the lungs and the systemic circuit through the organs and body. That's a pretty long definition, but it says most of it. To add to that, I want to explicitly point out this pulmonary circuit is from the heart to the lungs, back to the heart, and the systemic circuit is from the heart to the head and body, and then back to the heart. Now, I've been saying that these are the two main circuits, and again, double circulation implies that there are two circuits, but there's actually one more part of the body that doesn't get the oxygen that it needs from these circuits, and that is the heart itself. So there's another smaller circuit, which is why we don't call it triple circulation, but there's another circuit called the coronary circuit. So yes, the heart itself, it has veins and arteries. It needs to be supplied with oxygen. Obviously, it's working very hard, and so we call this the coronary circuit, from the heart to cardiac tissue, and then back to the heart again. Now, I mentioned last time that we're not learning the names of all these arteries and veins in the body, so moving on to comparing and contrasting circulatory systems in different groups of vertebrates. So what we have been looking at so far, here's a review, is what we see in humans, but also what we see in mammals and in birds. This double loop circulatory system, this double circulatory system from the heart. There's the systemic circuit, there's the pulmonary circuit. This is everything that we have learned so far. This is true for mammals and birds. So mammals and birds have a 4-chambered heart with double circulation. Now, let's contrast this with reptilian circulation, or most reptiles. I won't bother you with the ones that are the exceptions, but most reptiles have circulation like this. So, okay, we've got a right atrium. We've got something that's labeled a right ventricle. It's going out. There's a pulmonary circuit. It's coming in. There's a left atrium. There's something labeled a left ventricle that's going out to the systemic circuit. This looks exactly the same. What's different here? Well, if you look closely at this right ventricle and at this left ventricle, you will see there is a place where the blood can mix, and it turns, it's drawn sort of purple here in this diagram. There is a structure called a septum. Are we at septum count like 3 or 4? We've seen a ton of different things called septa in this quarter, but septum always just means a wall. There's this structure called a septum that tries to divide this ventricle into right and left halves, but it doesn't do a complete job. Deoxygenated blood from the right side of the ventricle and oxygenated blood from the left side of the ventricle mix with one another, and that is very bad. When you mix the blood together, you end up sending oxygenated blood to the lungs, which is a complete waste of time. It already has oxygen, it doesn't need to get more. And when they mix together, you end up sending deoxygenated blood to the head and body, which is also a waste. It's supposed to be delivering its oxygen, but if it's deoxygenated, it has no oxygen to deliver. So again, this septum tries to divide and sort of minimize this mixing, but this is something that happens. Remember, evolutionarily, we're sort of working our way backwards here. We started with mammals and birds, which were, the most sophisticated groups of vertebrates. So we're going to see things get progressively, I guess, less efficient because we're going backwards. So I can describe reptilian, most reptiles, as having a three-chambered heart. Again, that ventricle is not completely divided, so we could say that it's a single ventricle with two different parts to it. There's a septum that only partially divides this ventricle. There's some mixing of oxygenated and deoxygenated blood. Bad, and this is still double circulation. There are still two circuits here, even though there's some mixing going on. Okay, amphibian circulation. Now, this is still double circulation. Look, we've got blood coming into the heart, there's the systemic circuit, there's the pulmonary circuit, but look at this. Right atrium, ventricle. There's not even a septum here trying to divide this into two halves. You've got deoxygenated blood from the right side and oxygenated blood from the left side, really doing a lot of mixing here in this ventricle. Some of it's going to the lungs, some of it's going to the head and the body, but there's a lot of mixing here. So this is even worse than reptile circulation. So amphibians have a real three-chambered heart. There is a lot of mixing of oxygenated and deoxygenated blood. That's still bad. Again, this is still double circulation. Despite all this mixing going on, there are still two distinct loops. Okay, our final group of vertebrates, if you remember the chapters on phylogeny here, is going to be fish. Oh man, so this is atrium, ventricle. This is not double circulation. This is what we can call a single circuit circulation, where there's one ventricle that in a single pump is going to send this deoxygenated blood to the gills. Again, we're talking about fish, so gills, not lungs. And then to the body and head to deliver the oxygen, and then back to the heart again, all within a single pump. This is not as effective as double circulation, but I mean, hey, it gets the job done, and there's no mixing of deoxygenated and oxygenated blood. So fish have a two-chambered heart with single circuit circulation. And again, we worked backwards here because we started with humans, but you can imagine evolutionarily a heart like this developing into having two circuits, but it's not completely divided, and then a septum trying to, prevent this bad mixing of different types of bloods, and then finally evolving into this form of the best version of things. So we did work backwards, but should make sense evolutionarily. Okay, that's it for the circulatory system. Now I want to move on to the immune system. Now, the entire immune system can be split into two halves: the innate immune system and the adaptive immune system. Let's start by talking about the innate immune system, or the innate immune response. So the thing with the innate immune system is that it's very fast. It has an immediate response to threats, very effective here. But the innate immune system is not very specific. It has, as we will see, limited specificity for certain pathogens. Now, pathogen is a big key term that we're going to see a lot in this chapter. The key terms define pathogen as an agent, usually a microorganism that causes disease in the organisms that it invades. The reason why this term is so vague, calling it an agent and saying that it's usually a microorganism, is because a pathogen can be a lot of different things. It could be a bacterium. It could be a fungus, a protist. It could be a eukaryotic parasite. It could be a virus. That's why the definition says usually a microorganism. Viruses are not microorganisms, and so calling it an agent encompasses viruses as well. So yeah, we see pathogen a lot. It just means bad thing. Whether it's a virus or a bacterium or anything else trying to harm us, pathogen mean bad thing. So yeah, the innate immune system is not very specific, but it is very immediate. So the biggest part of the innate immune system is the largest organ of the human body, which, if you know trivia, the largest organ that humans have is the skin. Thank you, Google image search and probably moisturizer commercial. Yeah, the skin is a very effective barrier against pathogens. We saw a patch of skin before when we were looking at sensory receptors, but yeah, this epidermis, this outer layer of skin, is a pretty effective barrier, a physical barrier to entry. But beyond just being a layer of skin cells, there's actually a lot more going on in a patch of skin. A lot more going on. Don't sweat all the details here, although maybe the sweat this detail, the sweat. So the sweat that we make creates a low pH environment that makes it difficult for microorganisms to grow. Now, it's difficult for a lot of microorganisms to grow, but not impossible, because there are a lot of different species of microbes, bacteria, and some fungi that live on our skin. We call these our normal flora, and yeah, there's a whole ecosystem of living things in this patch of skin. They help to take up this valuable real estate and ensure that if some pathogens try to come in on this area, there's so much competition, it makes it very difficult for them to get a foothold. So the skin is a physical barrier to entry, an inhospitable environment for pathogens. This is due to the low pH of the skin and the normal flora, as they are called, that help to crowd out these pathogens. Now, the skin is great. It covers most of our body, but it cannot cover all of our body. We need entrances and we need exits. At these parts of the body, we have what are called mucosal epithelial surfaces at most of our entrances and exits. Again, the mouth and nasal passage and stuff like that. Cells that secrete mucus and enzymes to trap and destroy pathogens. Here's an example in the nasal epithelium. You've got these cells secreting mucus that, yeah, try to trap anything that may be trying to sneak inside the body through this opening. So, yeah, not much more to say here. Mucosal epithelial surfaces secrete mucus. Now, I talked about leukocytes in the previous chapter, the white blood cells that are part of blood. There are a lot of leukocytes that are going to be involved in the adaptive immune system, but there are definitely leukocytes that are involved in the innate immune system as well, and let's look at some of those. So some of these leukocytes are responsible for recognizing PRRs and PAMPs. So yeah, there's a lot of alphabet soup that happens in immunology in general, but let's walk through this process. So here's a typical leukocyte. It's called a macrophage. It has on its surface something called a pattern recognition receptor, or PRR. A pattern recognition receptor does exactly what it sounds like it does. It's a receptor that recognizes patterns. It's built to look out for common danger signals, common molecular patterns that mean bad thing, that mean danger, that means some sort of bacterium or other infectious agent, just some generically bad thing. Here's a bacterium with lipopolysaccharide. We didn't talk about this when we were talking about gram-negative bacteria, but it's part of their cell wall. It is a common danger pattern. What we call these common danger patterns are pathogen-associated molecular patterns, or PAMPs. So PRRs recognize PAMPs. And yep, here's the PRR of this macrophage, recognizing this PAMP present on a bacterium. Uh-oh, bad thing is present in the body. This is going to send out the alarm bells and release a bunch of cytokines, which all have delightful alphabet soup names like tumor necrosis factor alpha and IL-1 and IL-8, but it's going to signal for the immune system to get over here. There's something bad in the body. Let me summarize what I just went through. So this involves leukocytes. pattern recognition receptors, PRRs, on the surface of some leukocytes, macrophages are an example, bind to pathogen-associated molecular patterns, PAMPs, on common pathogens. Once these have bound, that leads to the release of these cytokines. This is an important key term. A cytokine is a chemical signaling molecule particularly important in immune system communications. So cytokines are really just ligands. Again, maybe you remember this slide from BISC1323. They're signaling molecules, they're ligands, but they're specific ligands that are involved in sending messages to and from the immune system. So now that we've released some cytokines, we've raised the alarm, this is going to signal for a different type of leukocyte to come in, a phagocytic leukocyte to come in and do phagocytosis on the pathogen. Phagocytosis should sound a little familiar to you. We talked about this when we were talking about protists. A lot of protists did phagocytosis as a mode of getting nutrition, of eating food particles. But animal cells can also be capable of doing phagocytosis. And so here is, an example of a leukocyte coming in, eating these bacteria, and then destroying them once they're inside of the cell. Now, I mentioned some of these phagocytic leukocytes. Neutrophils are a big one. They're really good at doing phagocytosis. And so are macrophages, which, okay, let's rewind a little bit. Hey, I said macrophages were the ones with pattern recognition receptors. Well, that's not a contradiction. They do both things. Macrophages are on the lookout for danger signals, but they also do phagocytosis, but with the help of neutrophils and some other phagocytic leukocytes. So this is how we get rid of bad things. This is how we get rid of pathogens floating around, recognize them, send out cytokines, bring in cells that will eat them, and then destroy them. This process can be brought about by, just bacteria floating around in the body, but this process can also be brought about through tissue damage. So when there's a breach of our main defense, again, the skin being our primary defense, when there's some some cut or something like that, all sorts of nasty bacteria and other pathogens are going to inevitably get underneath the skin through this wound, and that's going to release a lot of cytokines. These damaged cells are going to send out cytokines saying, hey, security breach, security breach. And here we see the leukocytes circulating in the blood system, again, the circulatory system. Remember, these are a component of blood. They're going to come out of the blood vessels into this interstitial fluid, along with these tissues, and they're going to phagocytize these bacteria. This process is called inflammation. So to summarize here, tissue damage can also lead to the release of cytokines and can also lead to phagocytosis. This tissue damage induces inflammation. Another important key term, inflammation is localized redness, swelling, heat, and pain that results from the movement of leukocytes and fluid through the opened capillaries to a site of infection. And yeah, if you've ever had a cut or something, you would notice redness, swelling, heat, and pain. A lot of that is because of these leukocytes coming in and doing their job. They're bringing a lot of fluid with them as well. That explains the swelling. That explains the redness. That explains the pain, as there's a lot of pressure of stuff entering this site, and explains the heat because a lot of the stuff moving in brings heat with it. So anyway, that is the process of inflammation. Now, this whole process does a great job of destroying external threats. Something has gotten into the body from the outside world. It's easy enough to identify it and to phagocytize it and get rid of it. But sometimes we also have internal threats that need to be dealt with. What do I mean by that? Well, let's do a quick sidebar. So, all cells in the body display pieces of proteins they have been making on something called a major histocompatibility complex, or MHC. Again, there's a lot of alphabet soup here, on an MHC class 1 molecule. Here's what this looks like. Again, every cell in the body is going to be making proteins, obviously. And all of these healthy cells, whether they're epithelial cells or whether they're muscle cells or whether they're hepatocytes in the liver, whatever they are, they have this MHC class 1 sort of pedestal here, and they're going to take pieces of the proteins they have been building and display them to the rest of the world on this MHC1. Hey, take a look at what I've been making. This is what I've been working on. Now, any cells that are virus infected or cancerous are messed up in a lot of ways. And a lot of times, this viral infection or the mutations that are associated with cancer lead to abnormal MHC1 or not having any MHC1 at all. We refer to these virus-infected cells or cancerous cells as altered self cells. Again, this is not an external threat coming from the outside world. These are our own cells that have kind of gone rogue and are doing things that they're not supposed to do. So these altered self cells may have messed up MHC1 or not have MHC1 at all. And here's an example of a virus-infected cell that is no longer producing MHC1. So it's not sharing its work with the rest of the class. It's not showing what it's been working on. So obviously we know there is something wrong here. End of sidebar. Now we bring in the leukocytes that are going to deal with this. They are called natural killer or NK cells, and they patrol the body searching for the presence of MHC1. Here's a healthy cell, has normal MHC1, go about your business. Here is a virus-infected cell, although this could also be a cancerous cell, that is not making any MHC1, or maybe it's making MHC1 that looks messed up. In this instance, the NK cell is going to induce apoptosis of this altered self cell. You may remember apoptosis from BISC1323. It's this process of programmed cell death. Every cell has a self-destruct button on it, and these natural killer cells are going to trigger this self-destruct button to prevent the spread of this virus or prevent the spread of cancer. So natural killer NK cells cause apoptosis in these altered self cells. Apoptosis, defined in the key terms, programmed cell death. Simple enough. And so again, this is not an external threat, it's an internal threat, but here we've got some leukocytes that help deal with this kind of problem. Okay, there's one more topic in the innate immune system before I move on, and it's something called complement. I'm sighing deeply at complement because complement is a very complicated cascade of enzymatic events that involve proteins cleaving other proteins and recombining and cutting other proteins and recombining. And so we're not going to do all of this detail in an intro course, but what I want you to know about this process called complement is that the complement system, again, it's part of the innate immune system. It uses a complex array of proteins that are present in the bloodstream that ultimately lead to lysis of the pathogen. That's what happens at the end of this cascade. There's this pore of proteins that forms inside of the pathogen cell and causes it to lyse or to burst open. So we'll return to complement. The reason why it's called complement is because it involves the innate immune system and it's going to involve the adaptive immune system. They complement one another, but for now, this is all I want you to know about complement. Bunch of proteins in the bloodstream that can be activated and lead to lysis of the pathogen. Okay, that's it for the innate immune system. Now we're moving on to the adaptive immune response. So the adaptive immune response is kind of the opposite of the innate immune response. The innate immune response was generic but fast. The adaptive immune response is slower but it's very specific. The way it works is it fights very strongly against specific threats as opposed to just anything that comes on the skin or lands in the mucus or has a common danger pattern. This is going to be very specific, but it does take longer to react. So there are several leukocytes involved in the adaptive immune system. Let's start by talking about B cells. B cells are responsible for what's called the humoral immune response. Again, this is still part of the adaptive immune system, but B cells are humoral immune response. That's just a phrase associated with them. B cells create antibodies. Very important key term, and antibodies are often abbreviated as ABS, just in figures and note taking and stuff like that. What is an antibody? Well, here's what it looks like. This is a protein, and it's kind of a weird protein. Again, I mentioned a couple chapters ago that most proteins are kind of globular, in generic terms. This is a distinctive Y-shaped protein. And let me read the key terms definition. An antibody is a large protein used by the immune system to identify and neutralize antigens. Antigen, there's another key term. So B cells create antibodies. The job of antibodies is to bind to epitopes of antigens. Okay, so an antigen is defined in the key terms as a substance, parentheses, protein, carbohydrate, etc., that an antibody binds to. So we're kind of in a circular logic here. Antibodies bind to antigens. An antigen is something that an antibody binds to. The reason we're so generic here is because antigens can be a lot of different things against proteins, carbohydrates, other small molecules. Antibodies bind to antigens. If an antibody binds to it, we call it an antigen. Okay, what's the epitope part of this? Well, when an antibody binds to an antigen, it's not binding to the entire protein antigen, for example. It's binding using this antigen binding site, the sort of tips of the Y here. It's binding to a specific region of the antigen. That specific structural region is called an epitope. So here's our generic protein antigen, but on this, there are all these structural regions called epitopes. And here we crudely rotated this antibody and see it binding to the pyramid epitope of this antigen. It's not binding the entire thing all at once. It's binding to a specific part of it. So the key terms define an epitope as a very specific structural region of an antigen. So you could say antibodies bind to antigens. You could say antibodies bind to epitopes. Those are both correct statements. Now, these antibodies, as I pointed out, have a distinctive Y shape to them, and they've got 2 antigen binding sites per antibody. That's what you get out of having this Y shape: antigen binding site, antigen binding site. Now, each B cell produces a unique antibody with unique epitope binding capabilities. They all have this same generic Y-looking shape, but there are tons of antibodies out there, and every single one binds to a unique epitope. So here's, for example, a bunch of different antigens, a bunch of different epitopes on these antigens. This particular antibody fits antigen A. It doesn't fit antigen D or E or B or C, or I guess it does fit A. It only fits A, not the other ones. These antibodies are very specific for what they sort of lock into and what antigen of what epitope they bind to. And each B cell only makes one specific antibody that binds to one specific epitope. Now, if we've got tons and tons of different antibodies, that must mean we need tons and tons of different B cells. That must also mean we need tons and tons of different genes, because antibodies are proteins. We know proteins come from genes. How do we get so many proteins? We must have a bunch of different genes. Well, that would be impossible. There's not enough room in our chromosome for 1 million different antibody genes. So in order to get this diversity, in order to get all these different antibodies, these B cells, as they're maturing, do something really crazy. They rearrange a portion of their chromosome, of their DNA, for the antibody gene as they mature. So don't get bogged down in the details, but there are lots of different possibilities in the genome for different structural regions of the protein. And yeah, there's some randomness to this of which parts get cut out and flipped around and rearranged into different combinations. Every B cell, as it's going through its maturation process, does this gene arrangement, and in doing so, it creates some randomly generated brand new antibody that is capable of recognizing some structural thing. So this is achieved by randomly rearranging DNA for the antibody gene in maturing B cells. So there are millions of different B cells in the body. Each one has a unique antibody gene, so there are millions of different antibodies present in the body. This is what makes the adaptive immune system adaptive. There are many possible antigens that it can recognize. So these are not just common danger patterns. By making this stuff randomly, if there's an antigen out there, your body probably has a way to respond to it. This allows the immune system to respond to many possible antigens. So how do these antibodies actually do their thing? Well, there are antibodies present on the surface of B cells, and if they bind to their antigen, that's go time. So, okay, let's look at this process. Here we've got a B cell and an antibody on the surface, and here it says antigen on bacterium. So yeah, okay, of all these millions of B cells in the body, this one has happened to find the one antigen that it was made to recognize. Yeah, that's absolutely go time. So it's going to present a piece of that antigen on its MHC2. Now, I just want to point out MHC1 was something that all cells have. We are not talking about MHC1 now. What's being presented here is a piece of the antigen that was found on MHC2. Not every cell has MHC2, just B cells and a couple of other leukocytes that we're not really covering, but these B cells specifically show their recognized antigen on their MHC2, Roman numerals by convention. So if an antibody binds its antigen, these B cells process it and present it on their MHC2. Who are they presenting it to? Well, they're not presenting it to natural killer cells. Again, that was an MHC1 kind of thing. They're presenting it to helper T cells. Now, we'll talk about helper T cells a little bit later in the chapter when we talk about T cells. Again, all these different things are sort of interconnected, so it's hard to talk about one without the other, but we'll talk about them more later. But what they're doing is they're sort of co-signing this. Like, hey, I've encountered this bad thing on my MHC2. Is it okay to proceed? The helper T cells are going to say, yep, it's okay to proceed. They're going to cause this particular B cell to clone itself. So remember, we've got millions of different B cells. Each one has a unique antibody gene and a unique antibody protein. They're all different from one another. But once this process has happened, this particular B cell clones itself. It divides, and all of these daughter cells are exactly the same. They have the same antibody gene, and they make the same antibody. They become what are called plasma cells or plasma B cells, which just means they secrete a buttload of antibodies. So we've presented this antigen on MHC2. Helper T cells recognize the presented antigen and cause that particular B cell to replicate and become plasma B cells. Plasma B cells secrete tons of antibodies. So now we have lots and lots and lots of antibodies being secreted, and they are all specific to this antigen that was actually encountered. Again, the adaptive immune system is a specific response. But what do the antibodies actually do? How does this Y-shaped protein help us? Well, there are a couple of different ways that antibodies can help us. One of them is they can neutralize viruses or toxins. So you remember from our virus chapter way earlier in the quarter, the first thing this virus has to do in order to infect a cell is attachment. It cannot attach to the surface of a potential host if it's coated with antibodies. Similarly, toxins like this diphtheria toxin, they also can't do their thing, whatever it is, if they're coated with antibodies. So having a bunch of antibodies against a specific threat helps neutralize these things. Additionally, antibodies can tag pathogens for phagocytosis. So, we talked about phagocytosis earlier. That was an innate immune system thing. But the thing is, these phagocytic cells aren't very smart. They can be brought into the site of infection by cytokines, but if they were to see some pathogen just floating around in the body, not on a wound site, they wouldn't necessarily know that it's something bad. However, if a pathogen is coated with antibodies, that's going to tell these phagocytic leukocytes, like the macrophage, oh, this is a bad thing. I'm supposed to destroy this. So that's what I mean by tagging these pathogens for phagocytosis. And the third thing that antibodies can do that help us out is they can activate complement. So they can tag pathogens to be lysed by complement. And again, this was part of the innate immune system, but we see it here in the adaptive immune system as well. These antibodies are helping these lysing pores of protein know where they're supposed to go. So they help make complement do its thing. One last thing I want to point out about antibodies. So I did say that they were all very specific. Again, this recognized, if I can go back to this recognized the A, but not the B, C, D, or E. Antibodies are specific for the epitope of the antigen that they bind. However, sometimes we find different antigens that have the same epitope or very similar epitopes. If these shapes are similar enough, we can get what's called cross-reactivity. Cross-reactivity, shown here, is, I'm reading from the key terms, the binding of an antibody to an epitope corresponding to an antigen that is different from the one the antibody was raised against. So let's say you had an immune response to this particular antigen with this weird, 3 spiky epitope. Now you've got antibodies that recognize this epitope. Well, here's a different antigen with a slightly different epitope, but it is close enough to where these same antibodies will also recognize the epitope of this antigen. And again, this can be a helpful thing if you have two different pathogens or two different strains of a virus that are similar enough. The antibodies you raise against one can help you fight off the other one. So yeah, just understand that this can happen sometimes. Different antigens with the same or very similar epitopes, you can get cross-reactivity with these antibodies. Okay, we are not done talking about the adaptive immune system, but as we're finishing up talking about B cells and antibodies, this is where we need to end things for recorded lecture 5-2. We'll do more immune system in the next one.